thanks for the extremely nice introduction. And thanks for the invitation to be giving grand rounds today. So, today I'm going to be talking about a platform that we established at the University of Colorado Anschutz Medical Campus, where I'm from. And before diving into the actual presentation, a few disclosure first. Funding and other disclosure that are not related to academic work and patents and NSBR funding. More important disclosure, I have a little bit of imposter syndrome when talking about transfusion medicine to an audience that knows about it much better than I. I'm a biochemist by training, so please bear with me if I'm not at times a little bit disconnected from the clinical translational relevance of some of the findings and more excited about specific molecular mechanisms. And the other disclosure is that throughout the presentation, I will be talking a lot about we did this, we did that, we observed this. When I say we, it's mostly people in the lab who actually get to do the work. In the last few years, I've become more of a scientific writer than, as most tenure positions unfortunately require you to do, than actually having fun in the lab with mass specs. And not just people in my lab, but a lot of collaborators nationwide and internationally. And for this very presentation today, I have to thank people in this slide and in this very slide. So this presentation is going to be made up of multiple sections. In the first part, I'm just going to introduce the technology that we are using at CU Anschutz. A little bit how I do really think that using mass spectrometry-based omics tools is really important to understand red cell biology, and our red blood cells are a perfect model to investigate oxygen stress responses in all sorts of conditions, responses to hypoxia or oxygen stress. And then in the second part of the presentation, I will specifically focus on red blood cell storage biology and the storage lesion and how we can leverage high-throughput omics approaches to perform large population studies in the field of transfusion medicine. So for those of you who have been or have never been at CU Anschutz, my lab is pretty much the whole floor at, so you can see my pointer here, the first floor of the Research Complex 1 South, and I have another lab, RC1 South and RC2 buildings on the same campus. And when I mentioned that we work with mass spectrometry-based approaches, I'm very lucky to have now 13 mass specs in the lab for all sorts of application. Since I believe this presentation is recorded, I'm throwing this very slide there showing all the capabilities or the most important capabilities from a proteomics, metabolomics, lipidomics standpoint that our lab can offer in the form of collaborative work. So why am I in love with red blood cells? Working in the Department of Biochemistry and Molecular Genetics, where the greatest majority of my colleagues focus their research on cancer and cancer biology. And yes, I get to do a lot of great work through colleagues at the University of Colorado Cancer Center. But my main interest is on red cells. And my colleagues in the department are like, why are you using these very expensive tools to investigate red cells? We already figured everything out, everything that was there to be figured out about red cells in the last 50, 60 years, so why bother? To convince them, I first start by saying that red cells are by large and far the most abundant cell in the human body, at approximately 5 million red cells per microliters in a 5-liter adult human individual, like 70 kilos, 175 centimeters tall. You have approximately 5 liters of blood. So that adds up to a total of around 25 trillion red cells in the human body. As for recent estimate, there's approximately 30 trillion host cells in the human body, excluding the 39 trillion bacteria, which adds up to a total of around 83% in the human body of cells being red cells, and around 90% of the total cells in the human body being blood cells altogether. Being a metabolomics person, being really interested in metabolism, red cells actually represent a simplified model in that they are devoid of de novo protein synthesis capacity because of the lack of nuclei and organelles. So their proteome is rather stable in that it is not impacted by de novo protein synthesis. And small molecule metabolites are actually the omics layer closer to the phenotype that can be directly assessed in the context of hypoxia and oxidant stress. Why is oxidant stress important to red blood cells? The main reason why everybody believes that red cells are extremely simple is because the majority of red cells, as the red cell, the erythroid precursor mature all the way to mature red cells, the very process of losing nuclei and organelles is necessary to make room for hemoglobin to transport gas, in particular oxygen, to be delivered in peripheral tissues constantly and particularly in the phase of hypoxia. This adds up to approximately 250 to 270 million copies of hemoglobin per cell. At full oxygen saturation, this adds up to approximately 1 billion molecules of oxygen per red blood cell, which is a lot of oxygen. Obviously, that oxygen is not just there, it's coordinated by prosthetic loop of hemoglobin with the heme and iron that coordinates it. But that much hemoglobin per red blood cells with these many red cells adds up to a total of 2.6 grams, approximately 2/3 of the total iron in the human body being stored in red cells. Now you have a cell that cannot make new protein, is exposed to, is loaded with hemoglobin, iron, and oxygen, and that can trigger Fenton and Haber-Weiss chemistry, induce generation reactive oxygen species. So it is a perfect model to study oxidant stress in the face of a cell that cannot make new protein to replace oxidatively damaged components. So I believe that it is the perfect model to study responses to oxidant stress, especially at the metabolic level. I also like to challenge the argument that red cells are really simple and we know everything that is there to be known about red cells. And I'm trying to do so by using another omics, proteomics, that was becoming more and more popular over the years. And our lab has particular expertise in blood proteomics. And even though 92% of the total proteome of our mature red blood cells is made up of a single class of protein, hemoglobins, it is also true that there's an additional, the remaining like 8% of the red cell proteome, membrane and cytosolic proteome, adds up to over 3,000 additional proteins that we can map against all the possible known enzymatic reaction in humans. And in red, you can see in this slide all the metabolic pathways beyond glycolysis and just branching here, the pentosphosphate pathway, which were the only two main pathways that were thought to be important to red blood cell metabolism, along with the Rappaport-Lubrin shunt that makes DPG, diphosphoglycerate. There's a lot more pathways that are potentially there, or at least the proteins that could exert the enzymatic activity to catalyze those specific reactions are there. And now we are trying to probe them one by one using high-throughput omics approaches to see whether the enzymes are not only there, but also active in a mature red cell. I mentioned multiple times that we use mass spectrometry-based approaches. The way we do that is that we lyse red cells, we separate hydrophilic and hydrophobic compounds. In this very slide in particular, I'm showing you a red cell hydrophilic compound extract. You have on the top right corner of the slide a 2D representation of a chromatogram, and on the center of these slides you have the 3D representation with an additional component being the mass to charge ratio, which is what the mass spec observes. Of course, the z-axis or y-axis for the 2D representation is the intensity, which is proportional to the abundance of a given metabolite. Each one of those peaks is potentially a named metabolite. As you can see, the majority of the spectrum is dominated by approximately 40-50 peaks, but you also have like little dots down here in what we in jargon call the grass, low abundant metabolites. And we can cut out 90% most abundant metabolites, and now we have something that looks like that with tens of thousands of, hundreds of thousands unique features, tens of thousands of unique chemical formulae that we then can attribute a name to based on the retention time, chemical physical properties. High resolution intact masses, topic pattern, chemical formula determination on the basis of those, and then validation against stabilized label internal standard or exogenous standard. And we have now libraries of small molecule metabolites, but also lipids. There is approximately 12,000 compounds in our standard library. One technological advancement that my lab has really pushed forward is the introduction of high-throughput metabolomics approaches. In particular, the development of methods that are 3 minutes long, 5 minutes long, 1 minute long, that are as sensitive, selective, and reproducible as standard approaches. And in the interest of time, I will not dig much into the details of that, but this has been highlighted by Technology Network and Nature. Springer Nature asked me to edit a whole thematic book that you can find online, and counter disclosure, I'm not getting any dollar out of this, so you're not doing me any favor if you buy this, but just if you're interested. I brought up the point that we can use metabolomics to probe whether those enzymes we find with proteomics approaches are not only just there, but also there and active. We can do that by using stable isotope labeled substrates, small molecule metabolites that are labeled at carbon atom through carbon 13, so stable isotope, non radioactive. And for example, add those compounds to a unit of blood and test a unit of blood on a weekly basis until the end of the storage period. And for example, and that's the reason why I believe that our list of over 3,000 proteins is incomplete, some enzymes like glutamate pyruvate transaminase that are not found in the list of 3,000 proteins that we and others have generated, the catalytic activity of pyruvate transamination to alanine is actually observable and quantifiable. We can actually use stable isotope label standards to generate literal Michaelis-Menten curve to determine not only that this reaction is actually there and active in mature red cells, but also determine how relevant that is in red cell and supernatants as a function of storage duration. And on the basis of all of this, we actually postulated that red cells could represent a sort of circulating organ. I understand this is kind of a provocatory concept, but the concept there being that if these enzymes, all of these enzymes are not just there, but also functional, red blood cells could actually take over some of the duties that are historically in textbook attributed specifically to other organs, such as, for example, transamination in the liver and so on and so forth. We can use all these approaches to investigate, for example, red cell role in response to oxygen stress, but also, for example, hypoxia. One representative study is, for example, investigating the role of red cell metabolic reprogramming in response to exposure to hypoxia at over 5,260 meters. Please don't ask me to translate that in feet. But long story short, in this study, we had a group of college students who had a paid vacation to Bolivia at over 5,000 meters. Little they knew that being carried to Bolivia on oxygen all the way to the top of the mountain and the oxygen being removed at the very moment they were at the top of the mountain would make them feel really, really sick, acute mountain sickness. So it wasn't really a great vacation for the first 24 hours. Good for them, all of them actually acclimatized really fast. And I will spare you the details that we published in multiple papers indicated down here. But in a series of 7-8 papers, relatively high impact, we ended up using a series of genetic and pharmacological models, genetic manipulation in mice and pharmacological interventions to define that in response to high-altitude hypoxia, both endothelial cells and red cells release adenosine triphosphate, and through a series of ectonucleotidases that results in increases in plasma adenosine, that adenosine signaling through adenosine receptor A2B, and ultimately impacts glycolysis by promoting biphosphoglycerate activity, biphosphoglycerate mutase activity. So these ultimately results in increases in the level 2,3-diphosphoglycerate, which promotes the stabilization of the tense deoxygenated state of hemoglobin, promoting oxygen offloading from hemoglobin. That deoxygenated hemoglobin can actually outcompete glycolytic enzymes that are otherwise bound and inhibited at the N-terminal cytosolic domain of BAN3. Those enzymes are now released from the membrane, migrate to the cytosol, and now they're more active, promoting kind of a feed-forward loop to make more DPG. An additional feed-forward loop that promotes tissue oxygenation. I don't think I have too many additional details here other than relevant to this presentation. This very mechanism can be observed through fluorescence microscopy and activity assays and stable isotope label tracers that show that GAPDH does indeed migrate in the cytosol in response to high-altitude hypoxia or ex vivo hypoxia, and that results in the activation of glyceraldehyde triphosphate dehydrogenase. How do we know that GAPDH does bind to BAN3? One way to probe that is, for example, to use cross-linking agent, in this case, disoxynyl subeate, and another one, DNTMM, that cross-links epsilon amine group of two lysine facing each other within 20 angstrom. If we use a zero-length cross-linker, we can use also other cross-linker that cross-links aspartate glutamase to lysine, and then just acidic group, carboxylic group of acidic amino acid to other carboxylic group of other amino acids. A long story short, create a map of the red blood cell interactome. So how these proteins interact with each other, or how you have intramolecular cross-link that are defining of the structure of a given protein in red cells. And pretty much redraw the maps of the red blood cell interactome, not just the cytosolic proteome, which is relevant, for example, to recapitulating decades of studies on TANS versus a relaxed state of hemoglobin, deoxy or oxyhemoglobin, but also, for example, to redraw the map of the red blood cell membrane cytoskeleton in the face of hypoxia and oxygen stress, which we found to be relevant in all sorts of conditions, from sickle cell disease to COVID, and unfortunately today I will not have much time to go into the details of that. You can actually go more targeted than that, use antibodies to pull down specific proteins of interest and do cross-links of the specific pull-downs, or you can recombinantly express, for example, an terminus cytosolic domain of band3 with specific flags that biochemically allow for enrichment through nickel columns, and then perform again these cross-links and go a little bit more granular on which residues of the N-terminus of band3 interact with which protein, including GAPDH. And again, you can do that with the SUMO tag or IS tag and perform pull-down and cross-links. And also, you can do other studies like thermal proteome profiling, where adding a given peptide of interest, in this case the N-terminus domain of band3, you identify all the proteins that either are stabilized in solution or precipitate in solution when the extracts are exposed to a gradient of temperature and are labeled with this technique that is called TMT, terminal mass tag, tandem mass tag terminal labeling. Ultimately, you can also recombinantly express the target protein of interest, in this very case GAPDH, with N-terminus cytosolic domain of band3, can cross-link the two, identify specific cross-link that inform not just that GAPDH does bind to band3, but how they do interact with each other, and ultimately perform computational modeling to predict this interaction, and isothermal titration calorimetry, 2D NMR and 3D NMR to determine the chemical shift perturbation, and ultimately map the very binding site of this N-terminus of binding domain of band3, which is otherwise intrinsically disordered and orders itself when binding with GAPDH. All of this introduction is to say there's a lot of omics techniques that we can use. We can use them to study all sorts of conditions. We have this specific project, and I apologize, it's kind of a self-serving slide. It's called Mirage, Metabolic Investigation of Red Cells as a function of aging, genetics, environment, and storage. If you have a project where you're literally throwing away red cells with a specific question in mind, we would love to run those red cells. We're getting samples pretty much from all over the States, South America, and Europe. And we use this to publish all sorts of papers. That's now over 370 something. And thanks for pointing out, John. Over 260 in the last 5 years. From studies with the Tour de France winner to COVID-19 to hibernation to high altitude hypoxia, hematopoiesis, and so on and so forth. Specifically blood storage, that's where we publish the most, which is the next and second half of the presentation. So when I started working on blood storage in the blood bank, I was a freshman PhD student in Professor Zolla's lab at Tuscia University in Italy. And we were approached by the newly established Italian National Blood Center that particularly read two papers. The first one was this paper, retrospective study by Dr. Colin Gormin-Cox, suggesting that there is increased risk of morbidity and mortality in patients in patients undergoing cardiac surgery, receiving units of blood older than 2 weeks. I will not have the time today to talk about all the issues that have been pointed out by many with the specific retrospective analysis performed in this study, but that was sufficient at the time to interest the Italian National Blood Center in understanding whether there was something that accumulates in the unit as storage progresses that may contribute to an increased morbidity and potentially mortality in transfusion recipients. To investigate that, we performed one of those studies that I was talking about, like getting red cells and throwing them on a mass spec. Red cell in this very slide stored in additive solution 3, but I will show some data from all sorts of additive solution. And as you can see in the slide here, from the top to bottom, you have an increase in storage duration from the freshest available blood to the end of the shelf life of packed red cells in the United States. Each single little dot here is a single biological replicate. At the time, we didn't have high throughput approaches, so we just did N equal of 5 per each time point. And this study took probably a month and a half or two. I'm bringing it up because I will show you by comparison how faster and many more samples we can run now. And what we did observe in this heat map is from blue to red, you have metabolites that accumulate over storage, and other metabolites that are consumed as storage progresses. So they are depleted from the red cells. And you can even almost draw a line in between storage day 7 and 14, suggesting that at least from a statistical standpoint, at the metabolic level, those red cells become something different. That, again, the clinical relevance and whether that storage lesion at the metabolic level can be reverted is a different story. That bar actually varies a lot depending on which storage additive is used. I will not have the time to go into details of this, but will suffice to say that we actually have a gradient of metabolic storage lesion depending on the additive solution with, and I'm not saying this because of Dr. S being here, but with SAGM in our hands at least being the worst, well, CPD being the worst, and alkaline additive solution, in particular additive solution 7 and Solex, being, to the best of my knowledge and our data, the best so far. If we really cursorily oversimplify the metabolic storage lesion in the blood bank, what happens is that the main energy substrate, glucose, is consumed through glycolysis. The main energy and only energy generating pathway in mature red cells is consumed as storage progresses, and this results in the accumulation of the end product of glycolysis, lactate, in mitochondria devoid red cells. Unfortunately, despite ongoing glycolysis, and there are some caveats there, but the high energy phosphate compounds, in particular adenosine triphosphate and diphosphoglycerate, are consumed almost completely in the case of DPG after storage week 2. This results in a red cell that is depleted of high energy phosphate compounds that promote oxygen offloading. So you have a hemoglobin that loads itself up with oxygen, and oxygen saturation increases as a function of storage duration. And concomitantly, if I convince you in the first few slides, you see accumulation of reactive oxygen species. Those reactive oxygen species ends up targeting proteins, metabolites, and lipids. For example, target, of course, the most abundant protein in red cells, hemoglobin, a functional residue, such as proximal histidine and cysteine 93 or 94 if you count N-hydromethionine as I did in this slide, which are relevant to gas transport, in particular oxygen, CO2, and some literature suggests nitric oxide. Is this relevant for oxygen binding and offloading kinetics? We've done single-cell studies on oxygen kinetics with Dr. Paolo Swiatek at University of Oxford, showing that tau and kappa, which are the two factors, the two values in the second scale range that indicate the capacity, the time to uptake oxygen, the time to offload oxygen, deteriorate with storage by alpha second over a second scale. We have some data that some of that can actually be rejuvenated with some specific intervention or even prevented with some specific storage condition, but I'm not free. I cannot share the data with you today. The paper has just been submitted. Of course, we can now not only look at things that change with storage, but we start correlating these phenotypes to clinical relevant covariates, for example, tau and kappa, capacity to offload and bind oxygen. I mentioned that hemoglobin is irreversibly and then irreversibly oxidized as storage progresses. Other proteins are targeted by this oxidative storage lesion, one being the very GAPDH that I talked about when talking about high altitude hypoxia. In particular, we see that asparagine residues facing the active site pocket of the enzyme and the very active site, cysteine 152, 156, cysteine 179, are reversibly first and then progressively irreversibly oxidized as storage progresses. It's actually an interesting mechanism that red cells use in the face of hypoxia to block late glycolysis. to promote a metabolic bottleneck and reroute glucose fluxes towards the so-called pentose pathway, which is rate limited by an enzyme, glucose 6-phosphate dehydrogenase, which is something I'm going to touch at the last part of the presentation. This pentose pathway generates a reducing equivalence, in particular NADPH, which is necessary to recycle oxidized glutathione and a series of antioxidant enzymes that are NADPH dependent and critical to red cell survival. While this phenomenon is really important to counteract oxidant stress, unfortunately it seems to fail as storage progresses, and the activity of GAPDH becomes irreversibly oxidized, irreversibly compromised by the end of the storage period. We can actually test the activation of the pentose pathway using stable isotope label tracers. I will spare you the details of isotope log determination or ratios here, but it will suffice to say that we do see that the cells try to reroute glucose moieties towards the pentose pathway. Unfortunately, that mechanism is not as productive in generating NADPH, or at least it's not sufficient to meet the demand for NADPH storage progresses. One of the reasons being that other factors that contribute to GAPDH inhibition, such as, for example, GAPDH binding to the N-terminus domain of BAN3, the very same mechanism that I discussed when talking about high-altitude hypoxia. If under hypoxic condition you have the oxyhemoglobin that binds to the N-terminus domain of BAN3 and displaces glycolytic enzymes to boost glycolysis, at high oxygen saturation you have the opposite. Oxyhemoglobin is now cytosolic, and GAPDH and other glycolytic enzymes bind to the N-terminus cytosolic domain BAN3 to promote pentose pathway activation by blocking late glycolysis at the expense of DPG and ATP generation. Hence one of the reasons why they get depleted as storage progresses. Alas, BAN3 too, on top of GAPDH, are target for reversible first and then irreversible oxidation through, for example, formation of azoaspartyl moieties that are progressively methylated by protein azoaspartyl O methyltransferase activity in a methionine and S-adenosyl methionine dependent fashion. Ultimately, what we have is that by the end of the storage period, a very highly oxygenated, almost full oxygen saturation red cell looks a lot like a deoxygenated red cells, where GAPDH is mostly cytosolic by the end of the storage period. We can actually test that mechanistically by generating mice that lack N-terminus domain of BAN3. The point being that if one of the reasons why storage is poor is because BAN3 gets oxidized and fermented, can we phenocopy the poor storage phenotype by making mice that are genetically ablated at the N-terminus domain of BAN3 at different residues, the ones that bind hemoglobin versus the one that bind GAPDH? And the answer is yes, and in particular the ones that bind GAPDH and the ones that bind hemoglobin have poor post-transfusional recovery, which can be in part prevented or at least restored, at least the metabolic defect, and I will show in the next slide the post-transfusion recovery, by incubating these cells from genetically ablated mice with cell membrane penetrating version of the N-terminus cytosolic domain of BAN3 that can scavenge and bind and inhibit GAPDH. And you can see here that you have the wild type or the GAPDH binding genetically ablated mice versus the hemoglobin binding mice that really don't have a response. So to summarize this fraction of the presentation, you have progressively oxidized proteins and lipids. You start vesiculating them in the supernatants, where you progressively accumulate, for example, irreversibly oxidized hemoglobin and GAPDH, and a series of other proteins. From left to right, you have storage duration. From blue to red, you have accumulation in supernatants. And ultimately, all this mechanism that I explained that requires activation of the pentose pathway to counteract oxidant stress at the expense of energy metabolism ultimately fails, and proteins and lipids that are oxidized are vesiculated in the supernatants, and red cells start to lose their viscosity phenotype by vesiculation, become acquiring an echinocytic phenotype, a spheroechinocytic phenotype, and ultimately a spherocytic phenotype, where the red cell maximizes the surface to volume ratio and becomes more susceptible to mechanic and osmotic hemolysis. In particular, by the end of the storage period, approximately more than 50% of the red cells are morphologically altered, and approximately 20 to 35% of the red cells are irreversibly morphologically altered. So they are echinocyte or spheroechinocyte, stage 3 and 4, and then ultimately spherocytes. What we can do is that by the end of the storage period, and this is in collaboration with Pascal Amirault and Pierre Buffet in Paris, we can actually label these endostorage red cells with specific fluorescence compound CFSE and use this labeling technique to sort the labeled cells, the one that look like this, horribly looking spheroechinocytic phenotypes, versus the one that are still discocytic red cells, even though at the end of the storage period. And then you can now sort them because you can label them and perform omics analysis and go the other way around. So you can actually ask the question, are the cells that are morphologically altered also suffering from all the metabolic and protein lesion that we observe as storage progresses? And the answer is yes. At the energy level, we see that you have extreme energy depletion, complete inactivation of the pentose phosphate pathway, and accumulation of oxidized glutathione and depletion of reduced glutathione in the morphologically altered, irreversibly morphologically altered cells, and accumulation of lipids, in particular phospholipids and peroxidized lipids, heats and odds, and depletion of the silk carnitine pool that is responsible for the damage repair of oxidized lipids in red cells. Of course, you can use all of this to generate a specific model red cell membrane. Actually use this model as, by feeding the model the lipidomics and proteomics data, and use this information to predict in silico the red cell bending modulus and the filterability of red cells by the spleen upon transfusion in vivo, which is indeed what happens in endostorage red cells that look like this. The morphologically altered, irreversibly altered red cells, the CFSE high, the one that labeled with our fluorophore and have all sorts of proteomics issues, they're actually the one that gets sequestered in post-transfusion recovery studies in mice, in post-transfusion recovery studies in humans, and in models of human spleen ex vivo perfusion. We can also go an additional step. We can label the red cells before transfusion. We can biotinylate them, and we can perform post-transfusion recovery studies on biotinylated red cells at the end of storage. But also we can recover those biotinylated red cells and perform proteomics and metabolomics analysis to understand whether and to what extent the storage lesion is reversed upon transfusion in vivo. And a good chunk of the storage lesion is actually indeed reversed in vivo. And again, admitting the survival bias, because the cells we can process, especially over time, are the ones that are not sequestered by the spleen. But we do observe that despite, for example, full recovery of ATP immediately after 6 hours from transfusion, there are pathways such as the pentose phosphate pathway, going back to the same pathway, antioxidant metabolism, that are not fully recovered even at 1 day from transfusion. Ribose phosphate here. So did we figure something out that is relevant to clinical transfusion practice? A few prospective clinical trials came up over the years, in the last few years, suggesting that, alas, for molecular mechanistic studies, the standard of practice seems to be non-inferior to selective transfusion, the freshest available blood. So if you were with me that day when this data were presented at ABB, this was pretty much the reaction of everybody, right? We don't need to change anything. Everything is fine, the field of transfusion medicine. Let's stop doing these studies that scare people away from blood. Blood saves lives, who cares? Why you keep doing this? This horrible science should never be funded. I do believe there is something there, and I do believe there is a disconnect between the basic science studies and these clinical trials, and I think it's some biology that is really relevant. One being, for example, that not all units store and recover the same. In classic study by Dumont and Aubuchon, data shows that while the majority of donors store and recover after 24-hour post-transfusion, more than 75% of the red cells you transfuse still circulates. But there are some donors that actually have very low post-transfusion recovery. You can actually phenocopy that in mice, where different mouse strains with different genetics, beautiful, elegant work by James Imring, now University of Virginia, formerly here in Seattle. Different mouse strains with different genetics store and recover differently. I will not have the details to go into the mechanisms that we found out for this. It will suffice to say that there is biological diversity, at least at the genetic level, that can contribute to explaining interdonor heterogeneity in blood storage quality and post-transfusion performances. To put it differently, blood-like people might not always age the same. Some people age really well, some people not necessarily so well, in this respect. And the age of blood in terms of days elapsed since the time of donation, and this is not a political statement, may not necessarily be relevant with respect to the quality of the transfused blood. So in other terms, the metabolic age and the chronological age of the unit may not necessarily overlap. In the next few minutes, I would just like to go through how we can use high-throughput omics approaches now to try and tackle the issue of genetic heterogeneity or even blood product heterogeneity as a function of population studies, as a function of donor genetics or other factors that may impact storage quality. To do so, I will leverage a cohort actually 4 cohorts of blood donors, healthy blood donors, enrolled at 4 different blood centers across the United States, project recipient epidemiology and donor assessment study led by Dr. Mike Bush at Vitalant. In this study, approximately 13,400 healthy donor volunteers were enrolled to donate a unit of blood that was stored for 42 days and tested for spontaneous hemolysis, osmotic or oxidative hemolysis, meaning the propensity of those red cells to hemolyze following osmotic or oxidative cells, and then they were fully genotyped. In addition, these are just the parameters that they use, the pink test for osmotic fragility and this AAPH test, which was a proxy for red cell propensity to hemolyze following oxidative salts. And here you have the distribution of hemolytic propensity in each one of these phenotypes. You see that you have different distribution depending on the phenotype. And in particular, you can identify the 5th and 95th percentile lowest and highest hemolyzer, ask them to donate a second unit of blood, store that unit of blood again for 42 days, and see whether this phenotype recapitulate intra-donor and across multiple donation. And the answer is yes for some parameters, no for others. So there are specific donor-dependent components at the genetic level, but also at the metabolic level now that we can only see because we are studying now populations that are larger than n equal of 5 in our initial studies. Now we can delve into, for example, the impact of sex, the impact of aging, the impact of body mass index, testosterone replacement therapy, BMI, other factors that I don't have the time to go into the details of that today. We can actually generate a portal that is going to be available online soon that is called the Mirage portal, namesake after my R21 and one of the other ones. And long story short, this portal will be accessible. You can go online and download all the data or just process the data real time, for example, as a function of additive solution, as a function of donor age, as a function of donor sex, and identify that, for example, there are sex-specific differences in creatinine. Creatinine was transporter is coded by gene on chromosome X, makes a lot of sense that it's sex dimorphic. But also, in classic studies, was associated with younger versus older red cell population, in that the creatine transporter gets lost from red cell membrane as red cell age. So if you have an average younger red cell population in circulation, which happens to be the case in premenopausal women, you have higher levels of creatinine. You can sort the red cell population with density gradient or other studies that I don't have the time to talk about today, and see that, for example, creatinine is actually indeed higher, elevated in young red blood cells from female donors at the beginning of storage. And that corresponds with red cells that are more deformable, less likely to stress, to respond, to hemolyze following oxygen in cells, and with higher capacity to carry and offload oxygen. And from a redox standpoint, also more resistant to oxygen stress. You can use all this wealth of metabolic information to correlate with the genotypic data and identify what we call MQTL, or metabolite quantitative trait loci, trying to understand which metabolite is linked to genetic heterogeneity, polymorphism across the blood donor population. And this is just from 250 donors in a pilot study. We just finished 2,000 blood donors, and we're now about to receive another 14,000. We have 10,000 in the freezer waiting for the last 3,000. Imagine how we can identify genetically encoded metabolic variants across blood donors. One such factor is, for example, G6PD deficiency associated with alterations of redox metabolism. The most common enzymopathy in humans affects the capacity to activate the pentose phosphate pathway. It's a trait that has been kind of fixed and spread across human population, impacting around 600 million people around the world, 500 to 600 million, depending on the review in the literature. It's very common in the African American population. Approximately 13% of African American blood donors in metropolitan areas like New York suffers from some degree of G6PD deficiency. And we can look for G6PD deficient donors in the rats population and hypothesize that since these individuals cannot activate the pentossustate pathway to counteract oxygen stress, their red cells should store worse, poorly. And indeed, that's what happens. During storage, G6PD activity declines for all the reasons that I explained in the first part of the presentation. In G6PD deficient subject, that activity is actually already med- variant, less than 10% of the residual, or less than 1% of the residual activity in the original healthy G6PD in fresh red cells. And that corresponds to all sorts of alterations of the pentossustate pathway and antioxidant capacity, and corresponds to alterations of red cell energy metabolism as well, because if you have excess oxygen stress, you cannot keep glycolysis going. And ultimately, all of this corresponds with decrease in post-transfusion recovery in blood donors with G6PD deficiency. I'll spare you the details of this, but we actually have now a mouse model that we can use to recapitulate all this information and perform mechanistic studies in. I mentioned multiple times that we have post-transfusion recovery metabolomics data. We can now use metabolomics and post-transfusion recovery or hemolysis to identify metabolic correlates, not just to functional measurement like oxygen kinetics, but also functional measurements like post-transfusion recovery, the capacity to circulate, or the capacity to hemolyze upon transfusion. And ultimately, the last 3 slides, we can focus on donor-specific factors that are not necessarily genetically encoded. And that's the beauty of metabolomics. You're closer to the phenotype. What are the factors that are not genetically encoded, such as, for example, smoking? We have markers of cotinine, nicotine, cotinine oxide, markers of whether the donor smokes or not, ethyl glucuronide, exposure to alcohol, for example, alcohol consumption, coffee. I'm at the 2nd or 3rd double espresso today. My blood will store better. Because the pentossustate pathway is apparently basically overactivated in response to caffeine, which is actually an adenosine receptor A2B agonist, going back to the studies on high altitude hypoxia. But then all sorts of additional small molecule metabolites that are, for example, over-the-counter drugs that are not ground for donor deferral, blood pressure medication, anti-inflammatory drugs, acetaminophen, paracetamol, or ibuprofen, statins. We see all of this in blood products that are routinely donated, and we're able to assess that now because we're studying samples in the thousands, which we are not able to do when we have very well curated small cohorts of n equal of 5. And we see that all these factors are impacting red cell metabolism. We can actually be sure that those drugs are the ones we know because we can incubate red cells from healthy donors with all these drugs and see what is the impact of, in this slide, 1,366 drugs on the red cell metabolic phenotype upon incubation at 37 degrees Celsius for 24 hours. Not only that, but now since we know the correlates to functional phenotype, oxygen offloading, post-transfusion recovery, we can pick the modifier that increase or decrease the marker that associate with these functional phenotypes and maybe use those metabolites as an additive to boost glycolysis and pentossustate pathway by leveraging the very mechanism that I explained through this presentation. And you're actually doing that, and that's the final disclosure and final slide, to develop novel additive solution, and I'm trying to lure Dr. Hess to work together on this, with high throughput screening platform where we can test in 96-well plate format few thousand additive solution in a matter of days at a rate of 8,000 a week, and use all of this not only to recapitulate what happens in a unit, but also to potentially improve red cell storage quality based on these molecular mechanisms. And the final disclosure there, we actually got an SBR to fund all these studies in collaboration in part with the Hemanex, the anaerobic storage guys, and Sinopia Biosciences. And I apologize for the long presentation, and with that, I will be happy to take any questions. Thanks again for the invite. Questions?
So that was awesome, by the way. The proteome seems to, there's going to be a lesion in the proteome that predicts the metabolome. Have you been able to correlate any of the changes in the proteome since you've done those studies with these, what appear to be irreversible changes in the metabolome?
Yeah, the easiest one are the GAPDH1, for example, right? Where you have glyceraldehyde, the 3-phosphate dehydrogenase is right upstream to 1,3-diphosphoglycerate that fuels the Rappaport Lubrin shunt via biphosphoglycerate mutase. If the enzymes that makes the substrate for DPG generation is inhibited, then you have a metabolic bottleneck upstream. So either you fuel the pathway from alternative routes, inosine, for example, through pyrophosphoribolysis or phosphoribolysis of inosine into ribose phosphate and then the apoxanthine moiety, and that re-enters the late glycolysis through the oxidative phase of the pentosphosphate pathway, or you're pretty much like, I'll kind of find the politically correct way to say that, but there's no way around that, right? The other option is to prevent that lesion in the very first place, using antioxidant, using alkaline dissolution, using anaerobic hypoxic hypocapnic storage of blood. There are different strategies around that. But the answer to that is yes, and we actually have evidence that if we target the proteome and specific proteins with genetic manipulation or pharmacological manipulation with specific inhibitors, we actually can accelerate the storage lesion or delay the storage lesion for given substrates.
Well, you can measure the storage lesion, and it does predict the outcome in the transfusion or not?
Well, we have no measures of outcome in the transfusion because we haven't done the studies yet. What I'm really excited about is in the let me stop using this. Okay. In the Red3 omics, these units from the 13,400 donors were transfused to recipients. So we're going to have data, the Kaiser Permanent Donor Recipient Link database, information on medical records, including hemoglobin increments, ICU-free days, and development of acute respiratory distress syndrome rather than chronic kidney disease, acute kidney dysfunction in these recipients, in the recipients of these units. 13,400 units. We can, for some of them, we can actually single out the single unit transfusion events and correlate metabolites and proteins to the specific outcomes. So far, we're limited to post-transfusion recovery, hemolysis, FDA and European Council gold standard for the quality of blood. I believe that they are necessary but not sufficient in determining the quality of the transfused blood. So factors like, for example, capacity to bind and offload oxygen are probably telling about function better than the capacity to circulate alone or not to hemolyze alone. But all these factors will compound to a biobank of information that we can feed systems to predict how blood is going to react in vivo and then test it prospectively in this cohort of 13,400 units, which we are, provided San Francisco sends us the last 3,000 samples, we already have 10,000 in the freezer. And it's going to be probably, maybe I'm an optimist by nature, but before summer, we're going to have the data. Please.
So I'm interested in the difference of those clinical outcomes for the cardiac surgery. Do you think now, knowing all of this about the metabolomics, can you go back and find a confounding factor or a latent factor in that original study that, did they recruit specifically from one type of donor? What do you think?
Well, that was a retrospective study. So they pretty much looked at everything they had that was transfused into recipients, and one of the confounding factors that the freshest available blood was preferentially transfused first, and the sicker recipients ended up receiving more units, and that increased their likelihood of receiving older units. So the sicker patients got also older units, and that was one of the confounders, right? To counteract, with what we know now, we have clear evidence that at least from a metabolic level and a post-transfusion recovery level, some endostorage blood looks better than some fresh blood. So some people should just be told, yes, it's great, you're saving lives, but probably your blood is good for 7 days. And some people, their blood should be stored for 63, 65, even longer, based on the molecular data. There are other factors, right? Plasticizers from the unit. what will happen when we'll switch away from DHP plasticizer to other plasticizer like DINCH or something that is deemed less toxic. Do we have to redo all of this, right? Good thing we can do it fast.
Dan Savitz asks, what's the median age of blood transfused in practice?
It depends on the region, and Dr. S will correct me. Colorado, 21, 28 days. Go to Alaska, where blood donation is, population is sparse. I've been told from the Colorado people that we're actually sending blood to California and Alaska when it is approaching expiration date, because we have more donors than they do, and we try and help them with shortage. There are areas like Italy where there are a lot of altruistic donors. The region where I'm from is a lucky one. Emilia-Romagna is another lucky one, where we always have excess blood and we send it to other regions in Italy where donation, unfortunately, the rate is not as high. So, Sicily, where it's summer pretty much all year long, and it's now, unfortunately, it's a piece of data that recapitulates in most countries. Around holiday periods, holiday season, people are less likely to donate blood or travel more, and which in some cases excludes them from being eligible for donation. So all these factors compound and impact in the average age of transfused blood. But that's a great point. In some countries, like in Europe, for example, Northern Europe, now the shelf life of packed red cells is 35 days. So you're also narrowing down the shelf life with potential impact on blood availability if and when there are shortages and mismatches in demand supply. The last 2 years have been a clear example of that with the COVID pandemic, and people either at first not being eligible or we didn't know whether COVID was transmittable through blood donation, and apparently the answer is no. And then also people ended up, the donors that were discouraged from donation at first ended up not donating anymore, at least in Colorado. I don't know if it is a shared issue nationwide. Thank you.
There are many other factors that also affect it. If a donor is hyperventilating when they're donating the blood, it will of course raise the pH, and the reducing capacity of glutathione falls very rapidly as pH falls. And so you can have the same level of glutathione, but it works much less well. It falls from like 0.7 of a volt at normal pH down to about 0.2 of a volt at about 6.4 at 42 days of storage. And this doesn't show up in your metabolic testing.
Well, because we didn't test on the basis of pH gradients, right? Yeah. And I don't have data on whether the donor at the time of donation was hyperventilating. What it does show, you have a series of other confounders. pH, among other things, is impacted, of course, by, free protons. But at the same time, you have parameters like lactate that are strongly impacting the pH status of the intracellular compartment. And you can use all of that to build back, like a kind of construct backwards an Nernst equation, right? to kind of figure out the redox status. What is a really good sensitive redox sensor for red cells is the glutathione status of the very cysteine 93 of hemoglobin. This guy here. It's approximately a 1-to-1 ratio between intracellular glutathione and intracellular hemoglobin. So in terms of molecules, hemoglobin molecules altogether are approximately 1-to-1 ratio with glutathione, meaning that full glutathione of this cysteine scavenges, sucks out all the free glutathione in the cell. And that could be our kind of canary in the gas light to determine at the time of donation the degree of basal oxidation of the unit. Of course, that is reversible, but to reverse that, you need reducing equivalents and a pH-dependent glutathione reductases. So you start already with an activation of a pathway, then other units only activates after week 2 or 3. Well, thanks so much for your attention. Hopefully it wasn't too boring.